So, good afternoon, everybody. Thank you very much, Dr. Poltekova from BioLab and OICR colleagues at large. So, this is a difficult task that I have been assigned with today. So, introduction into digital pathology, which I would say is impossible in one session, and you need more than one speaker to do that. So, I would say, first of all, that's an incomplete introduction to digital pathology, and maybe I would even rephrase it and say, okay, why go digital in pathology? So that would be a more appropriate title. And I realized that when I realized I cannot put that much information in 50 minutes or 1 hour, so you need perhaps you need an entire day to and multiple people to cover all aspects of digital pathology. But let's see where we go with this. So I have to disclose that I have several sponsors grants from industry, and I do consulting activities to disclose here before we talk. So this work and everything that we talk about it today with Amir Safarpur and Shivam Kaula is part of an Ontario government-funded consortium titled Computational Peer Review through Identification and Captioning of Gigapixel Digital Pathology Scans, which is supposed to go up to 2023. These are the core investigators of the consortium, including two pathologists, Dr. Batten from Grand River and Dr. Pantonovitz from University of Pittsburgh Medical Center. So Chemio Lab, so as was mentioned, is hosted at the Faculty of Engineering in WATERLOO, and we have at the moment several members, several partners, and we are entirely focused on identification and search in digital pathology archives, so we don't do anything else. With one exception, we have also a radiology project that Vector Institute has funded. So this is a challenge because the mixture of the audience does not allow you. We don't know really who is here and what we are doing in the mixture, but just as an overview. So perhaps the history of microscopy starts with the Nimrud lens, the piece of rock crystals that people use as magnifying glass, and the book of optics that shed light what is the relationship between optics and light some 1,000 years ago. So 1590 we got the early microscope, and Galileo Galilei gave us the compound microscope around 1600. So this is one of the reason that I think it has been neglected in literature. Why we are not going digital faster? I think that some of it has historical roots. We are using microscopes since 400 years, so it's really tough to put it aside. And the icon of microscope has become the symbol of science. So you take that away from a pathologist and you give it a monitor. That's not a replacement. So, but there is no literature work on that. So the first use of term microscope, 1625. First use of the term cells, 1665. So what you're talking about is actually quite old. And up to how we can image piece of tissue without staining it, for example. This is also not new. 1932 phase contrast microscope. Up to the recognition with Nobel Prize for different works on microscopes and the chemistry Nobel Prize for super microscopes. So is an old field, is a very established field, and it's really tough, among others, exactly for this reason to make any changes. So the modern pathology, we have many experts who can talk more detail about that today. So from our perspective, we look at the workflow and we try to understand what's going on. So patient goes to the hospital and a biopsy is taken and a tissue sample is taken. It goes to the lab usually and it gets processed. And after it gets processed, it goes to some sort of distribution channel, and that distribution brings the tissue sample to the pathologist, and the pathologist is the last instance. So what we do in radiology is basically an educated guess. Nobody gets treated based on what is suspicious in X-ray. So what the pathologist says goes. That's it. So that's the end of the line. That's the definitive diagnosis, and that's the reason that pathology is so important. And when the pathologist is done, he or she writes a report and files the report somewhere in the information system of the hospital and sends back the glass slides to the archive. And that piece of glass slides is everything. So is another major difference between pathology and radiology, that in radiology, you go in, you stay in front of the machine, and if an image is taken, not many of you, I'm guessing, remember the films. And it was really difficult early 90s to detach radiologists from the film. They wanted to, you would hear the argument, I want to touch the film. So why do you want to touch the film? So yeah, the image is on the screen, just look at it. But the fundamental difference of the pathologist is the glass slides is there, the block is there, it doesn't disappear. So it's a fundamental difference with radiology. And that piece of glass slides will go and get archived. Different countries have different rules, but basically we have to keep them for many, many years. The patient may come back, we have the block, we can go make another cut, take a look at it. Also, just not just for the patient record, but also probably for research. Although, to my knowledge, when the glass slides go to those large archives in the basement of the hospitals, usually they are not touched anymore. So they stay there, and most of the time nobody does anything with them. So, and it gets really complicated. And this is already, I show you an image that shows the actual complexity. And it gets complicated if pathologists are working on rare cases. Suddenly, then you have to add additional layer of complexity, that if I don't know what is on the glass slide, I have to send it back to the hospital. The hospital sends it to another hospital. Another hospital sends it to another pathologist. It gets packed up, barcoded, zipped, and so on, to get a second opinion from a subspecialist if it is something tough. So if it is lung adenocarcinoma, most likely we don't need to do that. That's a common case. There's a lot of expertise on that. But if it is some rare type of cancer, we need to do that. And that's one of the reasons that diagnosis get delayed. A lot of physical transport. So, so when we talk about virtual microscopy, so we have to go back to the microscopy. So there is nothing between the pathologist and the tissue sample but the microscope. So is confined in a space. But if you want to do that digitally, so now you have to put that glass slide into a machine, digitize it somehow, a computer, and then you can see it on a screen. And of course, for the actual microscopy, again, after you are done with the glass slide, it goes to the archive and stays there. So in the digital size, you send it to some sort of digital archive. So it's one of the arguments that digital pathology is better than conventional pathology because you don't need the physical space. True, but you need digital space, and you need massive digital space because pathology images are big. So it can be easily between 500 megabyte to 2-3 gigabyte, depending on the magnification. So showing you one image from the lab room, this is the complexity of conventional microscopy. I don't want to go in all details of that. We don't have time. I just want to draw your attention to all those manual physical transportations that we do in conventional microscopy. So whenever the manual distribution of the glass slides from the lab to the assigned pathologist, then we want to do archive it again manually, physically, you have to move it around. You want to do consultation, that's the nightmare. So because usually most of the time, the second opinion colleague is not there, is another institution. So you have to physically move things around. There are multi-billion-dollar companies doing just that. So when you want to change things, it's not just the mind of the pathologist that you need to change. Many of the pathologists are willing to do whatever it takes to be more efficient, but things have gotten established in the past 400 years, so it's not easy to change that. So if we move to pathology, to digital pathology as LabPoint themselves did, so you see that again, many things become digital. All those physical moving things around and transferring things get now suddenly converted into digital transportation. So you just send the image from here to here. Yes, that's 2 gigabyte pixel. It takes 30 seconds, but 30 seconds is still better than 2 days. So things get established fast. However, for those archives and servers, you need massive infrastructure. This is not something for the computer scientists or for the pathologists. This is something for the administration of the hospitals that they have to cope with. So yes, we know that's beneficial perhaps here and there, but who will pay for it to go digital? So you have to invest in that. And then the natural question is, what is the return of investment if I go digital? So storage-wise, this is, you need high-end, high-performance storage. You cannot just put it on HHD. You need SSD type of storage. So this type of things that could be at the same time regarded challenge or benefit of going digital. People are doing that. People are going 100% digital. And they report that if you go digital, you have efficient workflow, you have connected teams, you have increased safety, and of course, something that is not possible, new insights from analyzing massive number of images. So if I want to look at the relationship, which perhaps will be the holy grail of pathology, as if I look at the gene expression of certain diseases and correlate that with the morphology of the malignancy, what can we see? Well, nobody can say that because we don't have the tools yet. So we cannot make that connection yet, if there is a connection for at least some of the diseases. So there are things that would get better, but there are also things that we cannot do at the moment. So if you are looking for something specific, you cannot go in the basement of the hospital and grab 1,000 glass slides, bring them up, put them one by one under the microscope and show it to 10 different pathologists. You cannot do that. But in a digital context, this would be a matter of 30 minutes, full scale analysis. So of course, for digital pathology, you have to go and buy some scanners. There are several manufacturers on the market that sell different type of scanners. And there are type of trays and cassettes and glass slides that you can put. So you can go up from 1 by 3 inches, which is standard, up to 6 by 8 inches for brain sections. Basically, everything is possible. You can digitize any type of specimen. And when you do that, so one of the things is that you have to configure this. This is one of those things that changes the workflow in the lab, not necessarily for the pathologist. So maybe the histotech is the person who has to deal with this and say, what will happen to me or the lab technician when we go from conventional microscopy to digital pathology. So you have to learn, for example, to operate on this type of software, to set focus points, because the focus is not under the control of the pathologist anymore. So you are doing it before it gets to the pathology. This is one of the main concerns. So if I get the glass slides and I put it in the microscope as the pathologist, I can play and go back and forth between different magnifications as much as I want until I get and see the information that I need to see. But if it is software and digital pathology, that happens in the lab. The pathologist is usually not in the lab most of the time. The histotech is in the lab. So things will get shifted, workflow will change. And this is another reason for people to be cautious. Everybody wants to make progress, but the question is, okay, what would it take to do that? And after you get an image, then you have some sort of image viewer, image management system, then you can do usual stuff. This is where it gets really interesting, because now the tissue, the specimen is in the computer. Now the entire repository of computer vision and more recently artificial intelligence is available. Now we can do really nice things with the image, and hopefully we can take some of those burdens, laborious tasks that we should not assign to the pathologist. We can take it away and assign it to computers. Magnification is a point. I was just struggling up to yesterday. What in such a short time, what topic should I put here? Just when we talk about digital pathology, magnification is a sticky point. So because this is it, the pathology is about magnifying things, and when we go even 1,000 years back, it was always about magnifying things. So when we talk about magnification of in a conventional way, in conventional microscopy, is clear. We are talking about the eyepiece and objective lens. There's nothing in between, and the pathologist is in full control. So we can play with it to get it done. And magnification, the total magnification is clear. The magnification of the eyepiece times the objective magnification is your total magnification. So I don't know, medical physics 101 or physics 101. So that's clear. So when you go digital, things get shifted. So, and that's another reason that people are suspicious about digital pathology. Things that we got used to it for decades and centuries now are shifting, and we have to redefine them. Everybody has to be re-educated. We generally don't like that. If our knowledge gets obsolete and something new comes along, then okay, so then I have to go and find some new information. So magnification is not resolution. So that may be obvious, but we have to emphasize it at least for the digital case. The optical resolution is a function of the objective lens, whereas the digital resolution is a function of multiple factors. Is the objective lens of the camera, is the sensor of the camera, and is of course the monitor. The monitor is something new. So the monitor has not been there. So what is the effect of that? These are very serious questions that we ask if you're requiring pathologists to go digital. So how can I do that? There was no distance between my eye and the object going through the lens. Now there is distance when I look at a monitor. So what one of the good things is that you have added boost in magnification when you go digital, because you cannot really, you have to be really cautious when you talk about magnification for digital pathology, because you get the objective lens, but then you have the camera, and then you have the resolution of the camera, and things get magnified again on the monitor. So this is a boost generally, but I'm not used to it. I'm not used to see and work like this. So these are things that people are elaborating on and reflecting on to make a decision to go digital or not go digital. But there are three major concerns. Not money-wise. Again, administrators of the hospital will lecture us that, look, this will cost money. I cannot do that. We don't have that kind of money to completely change the IT infrastructure to. So average lab may do 700 patients a day, each one of them 2-3 glass slides, times 365 days. So that's terabyte and terabyte of data. So you need infrastructure to do that. We are not concerned with that. So from diagnostic perspective, what are the concerns? From the diagnostic perspective, everybody talks about three major things. There are other things, but they are not major, at least in my perception. So blurring, if part of the image is blurred, which was not the case before, this didn't exist, that the part of the image becomes blurry because I'm playing with the lenses myself as a pathologist. So I'm in full control of that, and I go back and forth, I get a really nice sharp picture, and then I see what I need to see. But in digital pathology, we don't know. And if the tissue sample is not even, then you may lose focus and things get blurry. And there was just recently a paper on what is the effect of blurry fragments of the specimen on diagnosis. Very serious issue, especially for rare cases where you may have a really in a tiny part of the glass slide being 100,000 by 100,000 pixels. Another point of concern is missing fade tissue. If the tissue is fade, maybe is, I don't know, some sort of, I don't know, it could be fatty structure, or could be some sort of special staining we do, ISC staining we do, and then certain structures get really low color, not intensive color, and then the camera do not pick them up if it is automatically done. If you don't manually insert those focal points, focus points manually for digital scanners, then you lose tissue. This has never happened. That freaks out every serious pathologist, that there was something on the glass slide that is now not present in the image. We cannot afford that. And this is one of the things that the companies, every single company who's in the field, they are working day and night to make sure this does not happen. Because if this happens, that diminishes the value of the digital pathology. But by far, the most serious concern is diagnostic equivalence. So if I diagnose a patient with based on an image and based on the microscopy, would that be the same? That's the most fundamental question. Doesn't matter that here and there may be discrepancies in the actual technology. So there are a lot of concordance reports that are in the literature. I just grabbed three that seemed important to me. So for example, you basically take 95 cases out of 100 and you say 100 cases and you look at, okay, I give it to one pathologist and say, okay, look, diagnose them based on the image. And then 2 weeks, don't do anything. Look at the same 100 patients again, this time diagnose them with a microscope. And then would we get the same result? 95% out of 100 in this study, the same result. And those 5 that was discrepancy were only slight discordance. So this is the type of studies that people do to make sure that the most important thing, would the diagnosis be the same? This is, if anything else is different, it doesn't matter. If we can make sure that the diagnosis is the same, then we should be okay. Another paper working on 2017 30 studies quoting percentage concordances, 95% or greater. 95% of the cases, digital pathology give you the same diagnosis as the microscopy. But also they say that some of them, they varied in quality, suggesting that further investigations, further studies are necessary. It could be that it's dependent on primary side and on the primary diagnosis. What is it that you're looking for? Sometimes the discordance could be higher or lower. We are not completely out of the wood, although the College of American Pathologists is making recommendations in that regard. Another one into, there are many. I just grabbed basically more or less randomly three that I like to show. So 694 live entire breast cases, concordance of 98.8%. Very impressive. Again, it depends. It depends. It depends what is the primary site you're looking at, how complex is the tissue, how complex is the manifestation of the malignancy, and then you may get lower or higher concordance. So again, the College of American Pathologists is making, this is one of the documents that is quite, you can download it at cap.org. So it's really, Dr. Parwani is one of the pioneers of digital pathology in US. So they make some recommendation, and three of them or four of them that I grabbed is, first one is this. So you have to evaluate the entire WSI, because you're replacing the microscope with a scanner, a computer, a display. So all those three components together are the replacement for the microscope. So you have to evaluate it together. And the CAP is suggesting that if you want to see whether digital is the same as microscopy, you have to look at least at 60 patients, at least. So, and they should be representative. They should be from the complexity and so representative. And you have to look at the diagnostic concordance. And now here is not important whether we agree or disagree with each other. In this case, the intra-observer variability matters. Same pathologist. What would he, she do if I give the pathologist this 60 cases? So 60 cases, and then most of the time we have a washout period of 2 weeks. So 60 cases, diagnose them today based on microscope, wait 2 weeks, same person, same pathologist, after 2 weeks, look at the 60 cases, this time digital images. Are they diagnosis different? One of the things that I think we should look at, and we have not looked at it, we know that there is inter-observer variability anyway. So you may repeat this just with the microscopy and you still get discordance. So microscopy after 2 weeks, same pathologist, you may have some variability in that. So how much of that can be assigned to the digital pathology? How much of that is to the inherent inter-observer variability? Still need to be worked out. But at least we have some recommendation after many, many years of, so based on the CAP recommendation, every hospital, every lab can make its own decision whether to adopt digital pathology or not. So that's really good. That's the result of many years of work. So
what's the benchmark for that? So what should be the concordance when you do the 60
the concordance, if I remember correctly, you can look it up in that page that I have added as a screenshot. The concordance should be above 94%, 95%. Then everybody's happy. Then we assign those 5% to maybe is inter-observer variability. So
why isn't it 100?
Well, because we are human, the perception is incomplete, things are incomplete, we get tired, we make mistakes, many other reasons. I don't think we can ever reach 100%, not just for digital versus conventional. I would argue in some cases even microscopy against microscopy, because we have inter-observer variability. Just put the digital pathology aside. So I'm sure you know there is a gigantic amount of literature on the variability, on the rate of variability, which is a problem in general in medical imaging. So we go digital from my perspective as a computer scientist because we want to use computer vision algorithms. That's why the way I look at it. So I look at the doctor, he's working or she is working, and I cannot do anything with those images because it's analog between the lenses and his eyes. So I'm useless. I cannot do anything as a computer scientist. So, but if it would be digital, I maybe also write some paper and get some grants. So that's my motivation. So blindly. But to do that, you need image data. That appears to be one of the obstacles right now in research, that you don't have image data. In Canada, the main factor for us is privacy protection. In Europe, more or less the same. In US, seems to be a little bit easier than here. In some spots of the world, there's basically no privacy protection. But among others, the NCI, National Cancer Institute, the TCGA program, you have large number of glass-slides, digitized, confirmed diagnosis with pathology reports, with genetic informations, if you like, so you can work with that. So there's a mixture of data. That's maybe the worst thing about it, or actually is a good thing about it, that it's heterogeneous. It comes from different manufacturers, it's not from one scanner, so that makes it good to really work with it. So this is one of the one source of data that we are using. Again, if you start from scratch and you're an average lab and you do 500, 600 patients a day as a small, let's say, community hospital, it will take a while to build up a data set. So, but there are publicly available data sets. So after you have the digital image, so what is it that you want to do with it? With computer vision, and more recently with AI. So of course, this is something that everybody is excited about. Sometimes we exaggerate with respect to the applications of AI. Most of the time, actually, many people exaggerate what is possible and what is not possible. But there are things that we can do. And the competency in computer vision and AI has been growing in the last 2-3 decades. Now things have become mature in some area of AI such that we can use it. So one of the things that we can use with digital pathology images, definitely we can classify them, and the absolute majority of papers are doing that. So, and that's fundamentally a yes and no decision. So yes, it is malignant, no, it is not malignant. So that seems to be very popular, at least with computer scientists and engineers. I'm not sure what is the value of that, and whenever I say that, I get criticized by my fellow computer scientists, engineers, because classification is 95% of what we do. So if you question the value of classification, so what is it that you are doing? So I don't like classification because at the moment we cannot provide the justification for classification. I can, after weeks of training of some sort of agent, I can just tell you yes, but I cannot say why. So that's a problem. Of course, we can also use AI techniques to generate images, basically fake images. And as much as that's disturbing in the personal domain, that you can, I don't know, make old paintings laugh and talk and so on. But in medical images could be a blessing. We can create synthetic images. That's a new way of creating phantom images. So if the science behind that is refined and we know what you're doing and there is no or limited possibility for abusing this, generating fake images is a new field that we can use to, especially if the label data is very expensive and we cannot ask our pathologists to generate certain samples for us. Computers could do that. So this is one of the things that, one of the fields that potentially computers can show that they can serve. They can be just a servant. They are not in charge, they just serve. And generating images is one of them. Of course, segmentation is an old thing, has been around for quite some time, with or without AI. You just look after getting a certain region, a certain tissue type, most of the time cell nuclei, segment them, count them, based on whatever diagnostic need you have. So it's also possible to search within archive of digital images. And this is the last one, is something that we focus on. The first three ones, and there are others, so these are the major ones. I'm not saying these are the only things we can do with computer vision and AI. So the first three are basically supervised techniques, which means you need the pathologist. Yes?
I was just wondering, in the classification you mentioned that justification is critical. I was wondering, how is the justification used downstream in the process?
So when you do classification, you cannot justify it,
Like
most of the time.
in the traditional or in non-digital pathology.
The non-digital pathology, the pathologist does it, and he or she writes a full-scale report, and he has to justify it, and he has to take the responsibility for it. So we have the pathology report. This is the justification. I look at it, and this is the, you describe it. I will show some example. And the supervised learning, again, so the problem is that you need the pathologist to come and sit down with you, not one, multiple, and show you, draw in the image, delineate some regions of interest and say, this is malignant, this is not malignant, this is a cell nucleus, this is not a cell nucleus, and so on. Of course, this is not practical. We are doing it on a limited manner. You see some of the public databases, you grab 200 images, two pathologists mark them, you break it down in tiles and patches, You run some experiment with patches, and then you publish a paper. We report 98% accuracy. Everybody's happy. Well, on the computer science, that's of limited use to actual pathology. The search is fundamentally unsupervised because you cannot train for search. If you train for search, you are doing classification. You are not searching. And perhaps search is a bit easier, but on the validation side is more difficult. One of the things that is interesting is stain normalization. So you go from lab to lab, and then depending on how you have your process of staining the specimen, things may be different. From day to day, it may be different in the same lab. So now, one of the applications of computer vision techniques are that you can take an image. The first one, for example, the template is you say, okay, you know what? So my image should look like this. The colorization of my image should look like this. And then the second one is you say, okay, the last one is the source. You say the first one on the left is the one that you say it should be like this. The last one on the right, the last column should be the images that you get. So, and the middle one is the one that you adjust. So you normalize it to become what you want, or you normalize it to a certain level. So you can create consistency. So what have we done with the color variation so far? Well, the pathologist has dealt with it like everything else. So if the staining has changed suddenly because we have a new histotag, or the H&E supply source has changed, or whatever. So that's the pathologist that has to deal with it. So, and he or she has to adjust his optics or understanding, pay more attention. Oh, this is very intensive. It seems that there are is a big structure. No, is two cell nuclei attached to each other because of the staining. Things like that. So now the computers have to deal with that. And one of the things that we do is a stain normalization. Of course, one of the things that everybody's talking about is deep features. So you take deep artificial neural network, which is just a collection of processing units, and you adjust their parameters with massive number of images. ImageNet is a collection of up to 16 million images that people use. So these are the images of cats and dogs and cars and buildings and bicycles. And you can, and this is a little bit freaky, you can use them so to train a network and then apply it on histopathology images. And at the colors that you see on the right, these are the activation maps for those histopathology images. Which part of that image made the network think that this pathology tile, this pathology patch belongs to this class or that class? So this is great from the research perspective, but I'm just waiting to see what happens if the first company or first institution brings this to the Health Canada or FDA and say, we train this with cats and dogs, and now we want to apply it on human tissue. So I don't want to be that person. So we are trying to train a network from scratch with histopathology images, which has its own challenges. One of the problems is you have to deal with unsupervised learning. If we cannot bug the pathologist all the time, please give us this, please give us that, then the value of unsupervised learning will be emphasized. So you have to work without label. You have to work without target, which parts of the AI community will assure you that this is the actual AI. The actual AI does not need even the target. The actual AI will go in and figure it out on its own. So for example, taking techniques like self-organizing maps, or even simple clustering techniques like K-mean, which are more than 50, 60 years old, and clustering the patches without knowing what they are. You don't need to know what they are. This is fat. This is epithelium. You don't need to know that. So the computer doesn't need to know that. The computer just need to know these are similar stuff and put them in one category. So, and then do something meaningful with it, which can then be interpreted by the pathologist. One of the things that the computer vision and AI are doing is the renaissance of old technologies like H&E. H&E is everywhere. H&E is cheap. We have automated the process. It's everywhere. We cannot, at the moment, we cannot, compared to ISC, which is more expensive, not fully automated, is highly selective. You need a lot of knowledge to do that. So if you look at the molecular testing versus H&E staining, when we focus on the first one entirely, so we risk losing valuable insight into histopathology, for example, the host stromal, reaction to cancer, and spatial relations such as tumor microenvironment and immune response to neoplasia. Whereas at the moment, most AI approaches that go after mitosis detection, segmentation, nucleus classification, and predicting Gleason scores have been using H&E, mainly because we have a lot of data. Maybe most glass slides are H&E. So I don't think H&E will go away so soon. So it will stay. It's practical. And now we are getting some value out of it. So why should it go anywhere? There will be cases that you cannot see whatever you want to see with H&E, and then you go with some biomarker, that's okay, and it will stay. And things will probably just get shifted. One of the issues is with AI, the reliability. So is it AI reliable? Can we rely on AI, or it will do stupid stuff? So it is possible that it does stupid stuff. For example, in this work, it was demonstrated that images of randomly sampled poses of turtles can be mistaken for rifles if you manipulate the network in a specific way that people call adversarial attack. What does that mean? Most likely, adversarial attacks will not happen in the medical imaging domain. Most likely. Well, if you make sure there is no cyber attack and nobody has access to the privacy information, then if the firewalls of the hospitals and research institutions are okay, then this will not happen. But what might happen is this, that some artifacts and noise may act as an adversarial attack, may trigger the same thing, and then suddenly lung adenocarcinoma may be labeled as prostate hyperplasia. Is that possible? It is possible. Nobody has observed it yet. People are looking into it. This is a serious concern. Again, direct adversarial attacks are not possible in medical imaging, but some other stuff, which we have many of them. We have artifacts left and right in the lab when we work with the specimen. Yes?
But don't we have the site information for the pathologist?
I would like to repeat
What do you mean site?
Sorry, one
Yeah,
second.
sure.
I would like you to repeat the questions using the microphone so the people from Sixth Floor can hear.
Okay, so there was a question. We will wait for the question. Okay.
Don't we have site information when the pathologist is receiving the digital images? Because this case, it's lung versus prostate. So
We
definitely there's
do, but if we give the full control to AI and AI can make some mistakes and it is not supervised, then things can go wrong. And this is something that definitely, even if we ignore it, regulatory bodies will look into that. So we have additional information. If somebody tell me lung adenocarcinoma is prostate hyperplasia, I will just point him to the barcode on the glass slide and say, just look it up in the lab information system. This is lung, this is not prostate. I don't need any recognition for that. But again, we are thinking ahead of time and say if we give full control or a lot of control to automated software, what can happen? And this is one of the things that can happen. And many, many serious people are working on this from different aspects to make sure we understand it fully. Pathology reports are something of immense value when you go digital, especially when you go digital. Looking at, for example, this thymoma case, it could be some cases recorded as tough case. Then you get the pathology report. The pathology report describes the entire specimen and says, this is a specimen, looks like this is the measurement, this is what I see, this is what I guess, and this is what I recommend. And in many, many cases, the language that recommends something is super cautious. Because don't forget, pathologists are scientists. As scientists, no scientist will make you assertion unless I know the sun is shining. This is the only thing that I can assert 100%. Otherwise, we talk always in a cautious language. And even when you get to the recommendation of treatment, you get super cautious. Say, yeah, I know this is thymoma. I recommend, it seems we have to do this, but this has to be discussed in the tumor board, for example. So we are very cautious when we do that. But what we can do with pathology reports, and many people have started looking into that, so just go through some natural language processing, get the keywords, get the phrases, can be recognized topics within the pathology reports. And again, I'm circling back to the NCI data with 33,000 glass slides and pathology reports, corresponding pathology reports. if we recognize topics, as if the computer is able to recognize topics with is more than keywords, recognize topics within pathology reports of evidently diagnosed cases, and we can find what does that mean with relationship to image. Now we will be opening new horizons. We are not there yet. Nobody is there yet, to my knowledge. So maybe in 10 years, we have to write many, many PhD theses to get there. But it is possible. And then what does that mean? We can automate the diagnosis of easy cases. Maybe in 10 years, we are there and the acceptance is there. Okay, so we understand, okay, the breast lobular carcinoma is not that difficult, let's say, and we can delegate it to the computers, and computers do 500 a day, and then the pathologists will come in and sign off. Look at them, yeah, no, the stupid computer had made a mistake here. So we need that transition to make sure that we gain trust in what computers do. What we have challenges when we process whole slide images, when you digitize the glass slides with one of those scanners. So one of them is that there is no label. The massive number of glass slides, digitized or not, they are not labeled, which means we don't know where is the suspicious thing, the thyroid cancer in this image. So if you don't know that, if there is no label, there's no way to use large number of computer vision techniques, including AI. So if I cannot do that, so what should I do if there is no labeled data? So again, the other venue is unsupervised learning. So can we go unsupervised? Other challenge is that these images are big. These images are gigantic. So everybody loves 4DX to look at the microenvironment, but then you have 100,000 by 100,000 pixel image. You cannot even load it into the memory at once. You have to look at it piece by piece. So we need to figure out techniques, fast techniques. And I was reading a paper, I will not mention from where it was, that was suggesting to do something with the images, pathology images, and 400 computers were used to get the task done. Well, good luck with getting that type of technology into hospitals in the foreseeable future, because we don't have that type of infrastructure. And I know what they would say. They would say, yeah, good, then buy some cloud subscription. Okay, somebody still has to pay for that. We have to get to a stage that we do things on a normal workstation. And we have to do a lot of work to get there. So, and of course, the major challenge in my mind is just the polymorphism, the gigantic, intractable diversity of patterns in the specimen. If you go, yes, we have only 4 tissue types, epithelium and nervous, and this, no, but if you go and depending on the primary site and all type of variations, there's sheer infinite number of tissue patterns. So there is not enough computational power in the universe, if even you had all those tissue patterns, to train something. You have to think about some other ways of doing it. We have also the dilemma of the feature extraction, which is how do we make the stupid computer understand the complexity of human tissue and animal tissue, for that matter. So yes, you grab a small part, a tile that you call a patch, because at the moment, no computer can process the entire glass slide at once. No one can do that. You have to break the glass slide into several thousand pieces and tiles and then process it. But when you do that, you get lost in the forest of pixels and then you lose the overview. So we cannot do that. But if you do that, generally you get, let's say, 1024 features at 32 bits. That's 4 kilobytes for every one of those. You have roughly for this image 4,500 patches. So it's 180 megabyte for this image. So I have already an image which is 800 megabyte, and you want to add 180 megabyte on top of it. That's one more reason for the administration of hospitals to not go digital, because then I have to buy even more storage. So as computer scientists and engineers, we cannot be generous with the requirements. I say, yeah, 400 computers and 3 major data centers to store your data, and then we can solve your problem. I would rather keep my pathologist. So, and don't go digital unless you give me another really reliable solution. So again, so we have real images, and then we have synthetic images, fake images. These are based on distribution, and they can be used for representation and simulation. So the research in that field has just started. So we do a lot of work on patch clustering, that the glass slice, the specimen comes, the digitized specimen comes, and we break it down in multiple clusters. Again, those clusters on the right, they don't have names and they don't need to be named for the computer. We just need to know this is class 1, this is class 2, this is class 3. What we want to do with that does not need to be labeled. And then you do your patch selection, you go through some sort of classical segmentation, you impose a grid on the image, you break it down in patches, and then you select the dependent patches, the patches that mean something. So you have to recognize redundancy, you have to recognize variability. And then you say, okay, it seems this pattern is repetitive. So whatever is everywhere probably is benign. Whatever is in lung and breast and prostate most likely is benign if you have a large database. If you're talking about 500,000 scans from all body parts, and you see a pattern in all of them, well, if it is everywhere, most likely is benign. Maybe it's fatty tissue. So, and everything that is special and is not very frequent, most likely is not benign. We are, again, talking from the perspective of the computer. So again, we could go on, and I want to save a little bit of time for questioning. So our work has been supported by several instances, as you see here. So I hope this condensed form, which is definitely not a complete introduction in digital pathology, has had some information, and hopefully, if you have questions, we can talk about those questions.